Have you ever wondered exactly how the microscopic instructions locked inside your DNA actually turn into the physical proteins that make up your body? It's basically a biological recipe book, where every single page holds the secret to life's machinery. So let's dive into this explainer and unlock that recipe book, unraveling the central dogma of molecular biology from gene to protein.
Here's our roadmap for today. First, we'll look at the blueprint of life. Then we'll break down step 1, transcription, and step 2, translation. Finally, we'll compare DNA and RNA and cover how to avoid those really common exam mistakes.
All right, part 1, the blueprint of life. Let's start with the foundation.
If you look at the left side of our split screen, you'll see a macro view of a whole cell. But as we zoom right into the nucleus in that magnified bubble on the right, we find our starting material, DNA, or deoxyribonucleic acid. It's the fundamental genetic material, literally containing the physical instructions required to build every specific protein your body needs. Check out the rungs on this twisted ladder. Notice how the specific nitrogenous bases lock together perfectly? We've got clear labels stamped right onto their corresponding rungs. Adenine always pairs with thymine, and cytosine always pairs with guanine. This highly specific interlocking mechanism is exactly what gives the DNA double helix its incredible structural stability. So what exactly is a gene? Well, if the entire DNA molecule is a massive recipe book, think of the highlighted segment you see right here as a single chapter. Within that chapter, every 3-letter sequence acts as a sentence, called a codon, and each codon codes for one very specific building block, an amino acid. The exact sequence of these codons is what entirely dictates your final protein structure. Moving right along to section 2, transcription, or copying the message.
Protein synthesis happens in 2 main parts, and as you can see on our flowchart, step 2 is dimmed out right now. That's because we're focusing entirely on step 1, transcription, a process that is physically confined within the boundaries of the cell's nucleus.
Here's where the action actually starts. See that enzyme labeled RNA polymerase? It's literally unzipping the DNA double helix. As it moves along the sequence, it reads the gene and builds a single contrasting strand of messenger RNA, or mRNA. Look closely at the labels. Notice how the new RNA strand uses uracil instead of the thymine we saw in DNA. The genetic message is actively being copied right before our eyes.
Next up, section 3, translation, where we assemble the protein.
Notice how our flowchart just updated. Step 1 fades into the background, and step 2, translation, is vividly highlighted. Just follow those thick vector arrows. The newly minted mRNA strand physically exits the boundary of the nucleus, passing through a pore, and moves right out into the cytoplasm for this crucial next stage.
Now we're at the construction side. Watch that structure called a ribosome clamp onto the mRNA. You can think of the ribosome as a master chef reading our mRNA recipe line by line. As it reads each codon, molecules called tRNA literally zoom in. Each tRNA brings an exact corresponding amino acid ingredient, matching the blueprint perfectly.
The absolute crucial point here is how these delivered amino acids physically interact. See those chemical bonds forming between them? They're linking together, one by one, into a growing sequence called a polypeptide chain. And once that linear chain is complete, it automatically begins folding into a complex 3D structure. Boom! That folded structure is your fully functional, finished protein. Section 4, comparing DNA and RNA. To really grasp this whole process, let's explicitly compare our 2 heavy hitters side by side. DNA features a deoxyribose sugar backbone and this thick double-stranded helix structure you see here. RNA, on the other hand, features a ribose sugar and a much thinner, single-stranded form. This physical contrast directly maps to their jobs. DNA needs the robust stability of a double helix to safely store information long-term, while RNA's single strand makes it a nimble messenger, absolutely perfect for carrying instructions to build proteins. Finally, section 5. Avoiding common exam mistakes. Understanding these mechanics is one thing, but applying it correctly under pressure is a whole different ballgame. So with all these interconnected variables and moving parts, where do students usually get their wires crossed? Look at the text callouts on screen, because these clear up the most common confusions right away. First, rule confusion. Never forget that DNA stores the info while RNA actively transcribes and translates it. Second, mixing up the 2 T's. Transcription is strictly the copying of the DNA to mRNA in the nucleus, whereas translation is the actual physical assembly of the protein out in the cytoplasm. Keep those action verbs firmly attached to their specific processes. Which brings us to our ultimate tip. Practice drawing and labeling these flowcharts and mechanisms yourself. Get a whiteboard, draw the cell boundaries, map the vectors of the mRNA, and label the RNA polymerase and ribosomes just like we did in this explainer today. By manually visualizing these steps, you aren't just memorizing facts, you're mastering the biological recipe book. And understanding how to read and rewrite that recipe book is the literal key to unlocking the future of genetic engineering, modern biotechnology, and maybe even the cures to humanity's greatest diseases. So, what will you do with that knowledge?